Hello there. This is Julie Huang again, a scientist in the San Francisco Bay Area and a volunteer of the Young Women in Bio program. I'm back to share what we know about testing for COVID-19 as of April 16, 2020. The science here is moving fast, so we will be back at a future date when there are more updates. But for now, let's get started with what we currently know. So what should you do if you think you may have COVID-19? First thing is to please stay home away from others if you think you're sick. Contact your doctor by phone or video or your local hospital if you do not have a doctor that you regularly see. There are now ways to have video chats with doctors known as telemedicine so that you can get information on how to proceed before getting to the doctor's office or hospital. This reduces the chances that you spread COVID-19 to others if you were to just show up and wait to be seen. If your doctor believes that you should be tested, which right now may still be difficult given the shortage of tests available, you may be asked to go to a drive-through testing center where nurses will get a nasal swab from you while you remain in your car. This lowers the risk of you transmitting COVID-19 to others as you don't have to set foot into a clinic or hospital for testing. The test involves getting a deep nasal swab from both nostrils, which you can see in this image. It's certainly an uncomfortable but necessary process. Because of a backlog in testing, you may not get your results until 4 to 6 days later. Though as new methods of testing are being implemented, the processing time for tests will be shorter. You'll need to isolate yourself at home for at least 14 days unless your symptoms become severe and you need to go to the hospital for care. Now, as I mentioned in the previous module on COVID-19, testing is extremely important to understanding how the disease is spreading across the community. Without knowing who is infected, we can never get a firm control over this virus. I'd like to now explain how exactly these diagnostics tests work. Diagnostics tests take advantage of what we know about the biology of SARS-CoV-2, the coronavirus behind COVID-19. As you may recall from the COVID-19 module, SARS-CoV-2 is an RNA virus. The RNA, a type of nucleic acid, is a unique fingerprint of the virus and can be used to identify it. The broad strategy of nucleic acid amplification involves using probes that can identify the unique signature in the nucleic acid and amplify that signal so that even very little viral RNA can be detected. One method, called RT-PCR, has been the main test used to detect the virus's RNA. The test can be done on the nasal swab samples containing viral particles, and it takes about 4 hours to extract the RNA, run the test, and get the final result. However, due to a huge backlog of samples that need to be tested and the need to ship samples to other facilities for processing, the turnaround time for getting results is taking about 4 to 6 days in some places. A new technology called ID Now, developed by the company Abbott, was recently approved by the FDA to be used to identify nucleic acids from SARS-CoV-2. Unlike the traditional RT-PCR method, this technology can obtain a positive result in 5 minutes and determine a negative result in 13 minutes. The new technology is also high throughput, processing many more samples at once compared to traditional methods. With the backlog the US has faced in testing, there is hope that with faster testing methods like the Abbott ID Now, the country can catch up to this virus. There is one other way to test whether someone has had COVID-19, and this test is based on what we know about the immune response to infection. When our body encounters a virus, our immune system quickly revs up to fight off this infection. Fever is a sign that our immune system is working hard to control the replication of the virus within our body. Eventually, if our immune system mounts an effective response to the virus, specific immune cells called B cells will start generating unique antibodies that recognize the virus. These Y-shaped antibodies, which you can see here in orange in this image surrounding a viral particle, binds the virus and helps to neutralize them. Even after the infection is cleared, these antibodies will remain in our bodies circulating in our bloodstream. So if a person has been infected with COVID-19 coronavirus, we can take a sample of their blood and do a test to see if we can detect antibodies against the virus. Because antibodies stick around even after the infection has resolved, the presence of the antibody doesn't mean the person still has an active infection. A positive result can mean that the person is still fighting off the virus, but is at the tail end of the infection, or they may have fully recovered and now have protective antibodies against SARS-CoV-2. Because this test is less likely to confirm an early active infection, the nucleic acid amplification method is the preferred diagnostic for COVID-19. However, antibody testing could help identify those who were infected but never developed symptoms and therefore may not have gotten tested using the nucleic acid amplification methods. This would help us understand how widespread the infection is in the community by including those who were asymptomatic. In addition, as more people recover from COVID-19, scientists are very interested in identifying the antibodies that are being generated against the virus, because this can aid in the development of a vaccine that can help protect those who haven't encountered the virus yet. Though testing is still limited, much progress has been made in the past month to make more diagnostics tests available to healthcare providers. This is absolutely critical as cases of COVID-19 surge in the US. However, it must be stressed that no test is perfect, and all diagnostics tests have a level of inaccuracy inherent to them. Two important concepts in testing to consider are rates of false positives and rates of false negatives. A false positive result means that someone who isn't infected is diagnosed as having the viral infection. A possible cause for this mistake is contamination of the sample with an actual sample that has coronavirus, or the test may not be sensitive enough to distinguish the RNA signature of SARS-CoV-2 with the RNA signatures of other non-COVID-19 coronaviruses. A false negative, on the other hand, is when someone is infected gets diagnosed as not having COVID-19. Possible causes for this inaccuracy is that sampling was not done at the correct anatomical location. For example, the nasal swab was not obtained deep enough to where the virus lurks, and so no virus was actually captured in the sample. Or it could be that the time of the infection when the sample was obtained ended up not being the right time to catch the virus, because the virus level or viral load varies over the course of the infection. Unfortunately, false negatives appear to happen more often than false positives. For example, in a Chinese study, about 30% of results obtained through RT-PCR may be false negatives. This high rate of false negatives means severe consequences as it underestimates how many are infected and inadvertently allow infected individuals to maintain their normal lives, thereby spreading the virus to others. As more patients are tested, doctors and scientists will have a better understanding of the accuracy of the tests they use. For now, doctors are advising that even if an individual gets a negative result for COVID-19, they should still act as if they do have COVID-19. While we have certainly heard a lot in the news about the devastating numbers of people who have died from COVID-19, there are a significant number of people who have fully recovered from this disease that we don't hear very much about. But tracking recovery is important as this will help the country determine when we may be able to ease social distancing measures. So how do we define recovery from COVID-19? The U.S. Center for Disease Control provides the following conditions that must be met to be considered fully recovered. You must have had no fever for at least 72 hours without taking fever-reducing medications. There must be improvement in other coronavirus-related symptoms such as no more body aches or difficulty in breathing. At least 7 days have had to pass since the first symptoms appeared. And lastly, if testing is available, it is ideal to have 2 negative test results that are at least 24 hours apart. Because testing is still not widely available, it is not mandatory to meet this last guideline, as it might be difficult to get 2 back-to-back tests done for one person. However, even if you have fully recovered, you should still follow social distancing recommendations so long as the virus is still spreading in the community. So, let's pause and review. What should you do if you suspect you have COVID-19? The answer is, first, inform your doctor of your condition. Second, get tested upon doctor's recommendation. Third, isolate yourself at home for at least 14 days. Fourth, if you test negative twice, you have fully recovered and are not contagious. And with that, I'd like to thank you for tuning into this module on COVID-19 testing. Here are some highlights that I hope you'll take away from this. There are 2 types of tests for COVID-19 currently available. One is a nucleic acid amplification assay that uses the nasal samples. A second is an antibody test, which uses blood samples. False positives and false negatives are inherent to any test. However, false negatives are more dangerous than false positives with COVID-19 diagnosis because it underestimates the true number of infected individuals. If you test positive, isolate yourself at home for 14 days unless you require hospitalization. Isolation can end once your fever is gone for at least 3 days and at least 7 days have passed since symptoms first appeared. But even after recovery, still follow social distancing recommendations. Congratulations! You now know the basics of COVID-19 testing and will be able to follow new developments as they occur. Please continue to check for updates and look for our next module on testing with more details because the new information is collected every day and this can change our current understanding of how to best diagnose and track COVID-19 infections. I hope this information empowers you to be proactive and take the proper precautions to protect yourself and your loved ones. Be well and stay safe.